Hello students, today let us learn about germplasm conservation. To start with the introduction, germplasm is defined as the genetic material which is transmitted from one generation to the next by means of the germ cells. It may also refer to an individual or clone which represents a type, species or culture and is kept in a repository for agronomic, historic or other reasons. On the other hand, the total variability present within all the living organisms and their ecosystems is known as the biological diversity or biodiversity. Biodiversity is commonly found at three levels, that is, occurrence of different environments in a system known as ecosystem diversity, the presence of different species reflected as species diversity, and genetic diversity or the different combination of genes within each species. Genetic diversity contributes the raw material for the production of different genetic combinations during the evolution of a particular species. These combinations of different genetic combinations enable the plant populations to face the challenges of changing environments like changing climate, new pests, new diseases, etc. Although human beings have realized the significance of genetic diversity from long time back, human activities are responsible for reduction in biodiversity, including genetic diversity. Such loss of genetic material in the form of genes or genotypes from individuals or populations is known as genetic erosion. Human activities such as clearing of forests, housing and industrial developments lead to genetic erosion. Moreover, the changes in cultural practices like the widespread use of a few standard varieties in place of the genetically rich traditional cultivated species also contribute to genetic erosion. Therefore, conservation of plant germplasm resources is important because it is required to save the resources from extinction, for enhancing agricultural productivity as well as to meet the needs of future generations. Let us now learn about the major categories of plant genetic resources. Plant genetic resources of cultivated plant species may be broadly grouped into the following categories. The first one are the wild species and weedy races. It represents the wild and weedy relatives of the cultivated plant belonging to the same genus or related genera. They are mostly found in the regions of true origin, which are primary centers of diversity and characterized by the presence of wild relatives, primitive characteristics and high frequencies of dominant alleles. The second are the varieties of traditional agriculture. These varieties of traditional agriculture include the land races, primitive cultivars and special purpose types. Land races are the early cultivated form of a crop species which evolved from a wild population, while primitive cultivars are the forms of crop species which are commonly developed from land races selected by farmers and cultivated under traditional agricultural systems without involving scientific improvement. On the other hand, the special purpose types are types originating from the areas of diversity and are adapted to specific ecological niches or provide special dietary or religious needs. The third are the products of scientific breeding programs. It includes modern cultivars, obsolete cultivars and other products of plant breeding or genetic studies like advanced breeding lines, stocks, mutants and gene markers. The modern cultivars include the high-yielding modern varieties like F hybrids, composites and synthetics, which have been selected mostly for their high uniformity and performance in intensive agricultural systems, while the improved varieties of recent past, which still persist as eco-strains in some areas, are known as obsolete cultivars. Next is genetic conservation. According to IUCN in 1980, conservation is defined as the management, preservation and use of genetic resources in such a way that they may yield maximum benefit to the present generation while maintaining their potential to meet the needs and aspirations of future generations at the same time. The major activities involved in sustainable plant germplasm conservation include proper identification, collection, characterization, evaluation, propagation, disease elimination, storage and distribution. Conservation of plant genetic resources requires concerted effort and cooperation of several disciplines and institutions and involves integrated and systematic oriented programs that may also need interactions of social, economic and ecological systems. Next, let us take up the types of conservation strategies. Conservation strategies are broadly categorized into two types: in situ conservation and ex situ conservation. First, let us see in situ conservation. It is a method of conserving germplasm in which germplasm is conserved in its natural habitats. Such type of conservation generally conserves the species along with their wild relatives, maintaining the genetic diversity and involves dynamic evolutionary conservation. Some common examples of in situ conservation strategy include national parks and sanctuaries, biosphere reserves, sacred groves, on-farm conservation and home gardens. National parks and sanctuaries are areas of land or water covering terrestrial, coastal or marine ecosystems which are protected to conserve the biological diversity and genetic diversity in its natural condition without human interference. In these national parks and sanctuaries, some eco-development programs are usually allowed around the protected areas with a view to maintain variability of populations and revitalization of the area without much harm from infrastructure development activities and introduction of new or improved varieties. A biosphere reserve is an area of land or water encompassing terrestrial, coastal or marine ecosystems which is protected by law of the state nominated by national governments, and they serve as sustainability support sites of genetic resources. The Man and Biosphere Program or MAB of United Nations Educational, Scientific and Cultural Organization or UNESCO was launched in 1971 with an aim to improve relationship of human populations with their environments with the help of scientific research and development. A biosphere reserve has three interrelated zones: a core zone comprising of a strictly protected part of the biosphere reserve that serves as the main site of conservation, a second zone known as the buffer zone including surrounding or adjoining areas of the core zone where some activities such as monitoring, scientific research, training and education etc. are allowed. And finally, transition zone, the part of the biosphere reserve where maximum activity is allowed for the local human populations to manage the natural resources in a socio-culturally and ecologically sustainable manner. Sacred groves are patches of vegetation protected by local human populations as part of their cultural and traditional religious beliefs and practices. On-farm conservation is the conservation of germplasm by farmers in traditional farms and agricultural systems, and such type of conservation is an easy and cost-effective way of conservation. Next, home gardens are also similar to on-farm conservation except that they are usually carried out in smaller scale. Next, let us take up ex situ conservation. Ex situ conservation is the type of conservation in which species are conserved outside their natural habitats. There are three main methods of ex situ conservation, namely seed banks, field gene banks and tissue culture. Germplasm collections conserved using any of these three methods are commonly known as gene banks. Germplasm materials conserved in these gene banks can be broadly divided into three types of collection. The first one are the base collection, which includes germplasm collection stored for long-term conservation. The second are the active collection, which includes germplasm collection stored below -150 degrees Celsius for medium-term conservation for about 30 years. And working collection, which includes collection of germplasm stored for use in breeding and improvement programs. Seed banks are the most convenient and efficient preservation method for conservation of plant genetic resources, since seeds are usually small and they are well adapted for storage. Most seeds are orthodox seeds, and such seeds can be dried well with low moisture content, and they do not lose their viability when stored at low temperature. However, some seeds are recalcitrant seeds and they cannot be dried and stored at low temperature without losing their viability. Field gene banks include active collections of germplasm in field or nursery conditions of plants which cannot be maintained as seeds. Next, tissue culture includes in vitro or in glass active collections of plant tissue cultures stored in slow growth conditions. Next, let us take up cryopreservation. Cryopreservation is another in vitro method for preservation of plant germplasm in liquid nitrogen at -196 degrees Celsius, where all biological processes are suspended. Some cryopreservation techniques include vitrifications or direct exposure of cells or tissues to highly concentrated solution of cryoprotectants, followed by rapid freezing leading to dehydration without the formation of ice crystals in cells. Next is encapsulation dehydration, which includes encapsulation of cells or tissues cultured in high sucrose concentration in alginate beds, followed by air drying and direct transfer to liquid nitrogen. And the next is two-step freezing, which includes incubation of tissue cultures or propagules in a cryoprotectant mixture solution for moderate cell dehydration, followed by slow freezing at the rate of 1 degree Celsius per minute till about 35 degrees Celsius before immersing in liquid nitrogen. Let us now see the conclusion. From the above discussions, it can be concluded that germplasm conservation generally encompasses interrelated activities of many systems and institutions. For efficient conservation of plant germplasms, a strategy of networking activities involving trained manpower is required. Gene banks are now emerging as preferable plant genetic resources centers. To reflect, it is more than just a storage place, takes care of a bulk of the germplasm conservation centers, and long-term maintenance of the gene banks is expected to serve the requirements of current breeding programs as well as provide the needs for future. Thank you.